Hello, everybody, and welcome to another episode of Silence Abounds, Science at Home, where we will discover home lab magic. Welcome back. If you're a new viewer and this is the first time watching the channel, welcome, and I'm very happy to have you watching my video. My channel is about basically I demonstrate scientific principles, reactions, and concepts by experiments that I perform in my home lab, covering chemistry, biology, microbiology, microscopy, and even molecular biology. So if this is your first time watching my video, again, welcome. And please, if you like the video, hit the subscribe button, hit the like, and please send me a feedback comment. I love to hear from everybody. In today's video, I am very excited to be able to bring to you and to demonstrate the CRISPR-Cas9 molecular biology gene editing technique. Yes, you heard me correctly. We are going to perform a CRISPR-Cas9 reaction. CRISPR-Cas9 is used to modify an organism's DNA at a precise location. To do this, a short customizable guide RNA is used to target a Cas protein to a specific DNA sequence. CRISPR-Cas9 is a genome editing system, originally evolved in bacteria as an adaptive immune defense system against viruses. Scientists repurposed it as a powerful tool to precisely modify DNA in living cells. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. Cas9 stands for CRISPR-associated protein 9. It's a DNA cutting endonuclease. In this lab today, we will use E. coli. Normally, E. coli will not grow on media containing streptomycin. Streptomycin binds to the ribosome and prevents it from making proteins, stopping the bacteria from replicating and growing. We will make a specific mutation in the ribosomal subunit protein that prevents streptomycin from binding it, allowing the bacteria to grow on the streptomycin media. It changes a single DNA base so that the lysine amino acid at position 43 is turned into threonine. The genome of the E. coli bacteria that we will engineer is over 4 million DNA bases in size, and CRISPR will find the single one that needs to be mutated. So the first part of the experiment is to grow our E. coli bacteria on LB media. So let's do that now. Okay, the very first step in this CRISPR experiment is to prepare the plates, the culture plates. And I need a plate that has LB agar to grow my E. coli bacteria. And then my second plate which will contain streptomycin and kanamycin in LB agar, and that is used for the final step of the experiment to show the transformation and the CRISPR reaction taking place, because normally, remember that E. coli doesn't normally grow on plates with streptomycin. But with this CRISPR experiment, we are going to manipulate the bacteria in order for it to grow and develop on streptomycin plates. So 2 plates I'm going to make today. The first is just a simple LB agar plate. So I have my agar and I'm going to add it to this glass container. It was pre-measured agar. And then what I want to do is then add 150 mLs of water. And I've got this measuring tube that I'm going to use. I could use a measuring cylinder, but if you don't have a measuring cylinder, you can use one of these plastic measuring devices. So I'm going to measure 150 mLs. So that's 3 lots of 50. Okay, so that's 150 mLs of water added. So what I'm going to do is shake that up and mix up all the agar. Make sure it's dissolved. Give it a vigorous shake. And now what I want to do is I want to boil. I have a hot water bath above boiling water here, and I'm going to put this, I'm going to loosen the cap. You don't want to tighten the cap. You just want to put the cap straight on like that, and I'm going to take it from the neck of the bottle, and I'm going to place this into my water bath, like so. I got a little bit too much water. Let me just pour off some of the water so it doesn't overflow. I have another beaker here. So I'm just going to pour some of the water out. Okay, that should be enough. And I'm going to put my bottle in of agar, and I'm going to let that dissolve. You don't exactly really want it to boil, the agar, but you want it hot enough to dissolve the agar and become translucent. When the agar has fully dissolved into the water, it becomes translucent, and the agar is then ready. So we're going to let that react, and we're going to let that dissolve, and then we'll come back when it's time to pour the plates. Okay, so the agar has fully dissolved and it has cooled down. So what I'm going to do is I'm going to pour the agar into my plate. Just going to hold it with a piece of tissue towel, and you just want to pour enough of the agar to cover the bottom of the plate, and no more than that. Okay, and that's all you need. And what I'm going to do now is just let the plate cool down for a couple of hours before applying my bacteria. I'm going to repeat the process for my other plate that I want, my streptomycin plate. I'm now going to prepare that and have that ready for the experiment. So now I'm going to prepare my culture, my E. coli culture. I have a non-pathogenic freeze-dried E. coli, and I'm going to reconstitute it with 100 microliters of sterile water, which I'm going to prepare now. So once I've added my water, it should reconstitute very easily and quickly. So I'm just going to give it time and flick the tube a couple of times, and this should facilitate the reconstitution. Okay, so now that's reconstituted, I'm going to now add the full contents of my reconstituted tube and apply it to my LB agar plate. So I'm going to pipette the contents of my tube onto the dish. And then I'm going to spread the liquid using an inoculating loop. I'm going to spread my culture, my bacterial culture, across the agar plate. Okay, so I've inoculated the plate. I'm letting it dry for a couple of minutes. And then I'm going to incubate this plate for 1 to 2 days at 37 degrees centigrade. And here is my plate in my incubator. Okay, so my LB agar plate has been incubating for a few days, and I have a beautiful growth of E. coli. So we move on next to making these bacterial cells competent. What do we mean by making competent bacterial cells? Preparing, basically, it means we are going to prepare bacteria so they can efficiently take up foreign DNA during transformation. We will use a transformation mix buffer, which I have here, and this buffer contains calcium chloride, which helps neutralize negative charges on bacterial membranes and DNA. Also in the bacterial mixture is PEG 8000. PEG 8000 is a high molecular weight polymer, and it creates a molecular crowding effect. This helps bring plasmid DNA closer to the bacterial cell surface, increasing chances of uptake. So before we move on to the bacterial transformation and CRISPR reaction, we need to make our bacterial cells competent. And so we're going to be doing that right now. So what I'm going to do first is add 100 microliters of the bacterial transformation mix into a microcentrifuge tube. So I'm going to add my buffer, which contains the calcium chloride and PEG 8000. And then what I'm going to do is I'm going to add some of my bacteria that I've grown using an inoculating loop. And you just want enough bacteria to make the solution cloudy, but still able to see through the tube. So I'm just going to take some bacteria and fill my loop. I want to have enough on my loop. Okay, I think I've got plenty of bacteria, and now I'm going to put it into my little tube and mix it into the buffer. And what I'm going to do is, using a pipette, I'm going to mix it up and down to disperse the bacteria cells, break down the cells. Takes a bit of time to break down. So I'm just going to keep doing this up and down pipetting motion until I think it's all dispersed. Okay, we are going to now move on to the next stage, which is DNA transformation and the CRISPR reaction. DNA transformation is the process by which bacteria take up foreign DNA from their surroundings and incorporate it into their cells. The steps of the CRISPR reaction include the Cas9 binding to the guide RNA molecule. Second, the Cas9 guide RNA complex finds the DNA sequence that matches the guide RNA. Step 3, the Cas9 cuts the DNA. Step 4 is the cell's repair machinery fixes the break using the supplied DNA template. And step 5, the cell has new DNA template. Cas9 works together with a guide RNA. The guide RNA directs Cas9 to a matching DNA sequence. Cas9 checks for a nearby PAM sequence, PAM standing for photospace adjacent motif. It's a short DNA sequence located next to the target DNA region that the Cas9 enzyme uses. Well, the Cas9 enzyme must recognize before being able to cut the DNA. If the match is correct, the Cas9 cuts both strands of the DNA. Once the Cas9 protein makes a cut of the gene we want to edit, the cell begins to try and repair. We will flood the cell with a template that is similar to the missing region but has a mutation. The cell will mistake it for a true copy and use it instead. Remember, our template has a single base change from adenine to cytosine in the ribosome. This change allows the bacteria cell to grow on streptomycin media. So during bacterial transformation, we add the Cas9, the guided RNA, and the DNA template. Once those are added to our transformation mix, We then incubate on ice and then rapidly heat to 42 degrees centigrade for about 30 seconds. And it's this sudden change in temperature that alters the membrane tension and opens up transient adhesion zones for plasmid DNA. So now let's perform the DNA transformation and CRISPR reaction. Okay, so I have my transformation bacteria mix in my tube here, and then I've got my plasmids, I've got my guided RNA, my Cas9, and my DNA template. So I need to add 10 microliters of each into my transformation mix. So that's what I'm going to do now. First is I'm going to add my Cas9. I mean, it doesn't really matter what order you put them in. So 10 microliters of Cas9. Okay. And then I'm going to add my guided RNA. Again, 10 microliters. Like so. And finally, my DNA template. Okay, so I've added all my reaction components and give it a little flick just to mix everything, make sure everything is properly mixed. And then I'm going to incubate this tube for 30 minutes on ice. So I've got a ice bath here. So I'm going to put that in my tube in like so and let that sit for 30 minutes. Okay, it's been 30 minutes incubating in ice. So I'm going to remove the tube and I'm going to put it straight into a beaker that is 42 degrees for 30 seconds. Okay, 30 seconds. We are done. And the next stage is I have a tube of LB media here. And what I'm going to do is I'm going to add 250 microliters of LB media to my competent cell mixture containing the DNA. Okay, so I want 250 microliters. So I've only got 100 microliter pipette, so I have to do it in stages. So I got 100, 200 microliters, and then I'm going to add 50. Okay. And 50. Okay, 250 of LB media added to my competent cell mixture. Going to give that a mix just by flicking the tube to mix everything up. And then what I'm going to do is this is going to be incubated at room temperature for 4 to 12 hours. So I'm going to incubate it overnight. And then the next day, I'm going to plate my mixture onto my streptomycin media plate. Okay, so now I'm just going to leave that to incubate overnight. Okay, so our competent DNA cell mixture has been incubating overnight. It's important that we give time and don't rush this stage because it allows bacteria to recover and replicate the DNA and perform the CRISPR engineering process. So you could have moved to the final stage after about 4 hours, but I wanted to make sure the reaction had enough time. So that's why I left it incubating overnight. So now we are ready to move on to the final stage of our edit bacteria with CRISPR experiment. And we are going to now apply our DNA mixture to our streptomycin media plate. So remember, this is what the whole point of the experiment was, to demonstrate that we could edit bacteria, the E. coli, with a mutation DNA template to allow E. coli to grow on streptomycin media, which normally E. coli cannot grow. So let's now perform the final stage and inoculate our streptomycin plate with our DNA mixture that we have here. So I've got my mixture here, and what I'm going to do is apply all of this mixture to my streptomycin media plate. So I'm going to pipette all of that onto the plate, and then I'm going to spread the liquid around the plate using an inoculation loop. So I'm just going to use a pipette, and then okay. And then what I'm going to do is get my inoculating loop. And then what I'm going to do is spread the liquid around the media and allow it to dry. And then I'm just going to incubate the plate for 12 up to 48 hours. I'm probably going to check on the plate periodically, but what I'm looking for is for bacterial growth to occur on the plate. So I'm just going to streak my plate with my inoculating loop to spread my mix nicely across the plate like so. And that should suffice. And like I said, I'm going to let that dry for a couple of minutes and then allow the bacteria, hopefully, if everything worked, I will start to see bacterial growth in 12 to 48 hours. So I will bring you back once I start to see bacterial growth on my streptomycin plate. So after a couple of days, I have noticed a successful CRISPR experiment reaction, and I witnessed whitish bacteria growing on the plate as seen here. These are bacterial colonies that were successfully edited, and so they survived and replicate to form small colonies, which are small groups of bacteria. So I'm very excited by my results. This was a successful CRISPR reaction demonstrating the technique. While I'm stoked that the experiment worked and I was able to demonstrate a CRISPR editing reaction in my home lab, and I hope you found that video interesting. So there are the gene editing technique of CRISPR itself has enormous potential in medicine, agriculture, and biological research. But it also raises significant ethical concerns. One of the most debated issues is the editing of germline cells, like sperm, embryo, eggs. Changes made to these cells can be inherited by future generations. Future generations cannot consent to genetic modifications made on their behalf. Unexpected mutations or side effects could be passed down permanently. Altering the human gene pool could have unforeseen long-term consequences. Many people support using CRISPR to treat serious genetic diseases such as sickle cell and cystic fibrosis. However, ethical concerns grow when editing is used for enhancement rather than treatment, such as increasing intelligence, altering physical appearance, enhancing athletic performance. This raises questions about what constitutes a disease versus a desirable trait. The debate is going to go on and on and on. CRISPR is just at its beginning, and the future is bright, but it has to be controlled and regulated. And there's going to be more on this as CRISPR develops and is used more and more. Well, thank you for joining me today for this video. Please hit the subscribe button. It means so much to me. Your support helps support the channel, keeps the channel going. Please hit the like button and please feel free to send me a comment. I love to get your feedback. Also, feel free to email me. My email is in the video description. And if you want the notes that accompany this video describing the scientific process and technique that we use today, please send me an email with your request and I'll be happy to send them on to you for free. Until next time, keep on experimenting. Goodbye, everybody.